Hello everyone, thank you for attending today's webinar on new polymers for transfection in vitro and in vivo. We are here today with Cédric Sapet, our technical specialist. We will start with the presentation and then we will host a Q&A session, and Cédric will be here to answer all of your questions. So I will launch the presentation and see you just after.
Biosciences is a French biotech company founded 20 years ago, specializing in molecular delivery systems. We have operations in both France and the United States. Our French site, located in Marseille, houses the company headquarters and production facilities, with 2 laboratories spanning over 500 square meters. Biosciences oversees R&D, production, our mRNA and LNP platforms, as well as marketing and sales. Our US site, based in San Diego, focuses on marketing, sales, and business development. Over the years, we've established a worldwide distributor network, with one of our key goals being to expand the reach of our reagents and expertise globally. Our expertise is built on more than 2 decades of research, innovation, and product development. Some major milestones include the launch of the first magnetofection reagents in 2004, the creation of OZB Inc. in 2013, and more recently, the acquisition of new facilities dedicated to LNP and mRNA platform development. We are proud to cover a huge variety of fields, from bioproduction to viral applications, going through in vivo protein delivery or stem cell applications. All the products developed and sold, as well as our platforms, are under the ISO 9001 certification. These innovative reagents allow Biosciences to offer solutions in 3 main areas: cell and gene therapy, bioproduction, and custom platform productions. And considering cell and gene therapy, we are glad to present you today our innovative solutions in Polyfection technology for in vitro and in vivo transfection reagents. Considering transfection of nucleic acids, several solutions exist, but even if they each present some interest, they are still not optimal. Electroporation or nucleofection are very efficient transfection methods that work well, especially for suspension cells, but they induce high toxicity and are less adequate to high throughput screening. Moreover, their cost is really expensive. Viral infection is maybe the most efficient way to genetically modify cells, and it allows long-term expression of nucleic acids. However, it is technically challenging, for the creation of the viral vectors, for example, impose hazardous handling and can be very expensive. On the other hand, physical methods such as microinjection or bioballistics show extreme precision and can be used for refractory cells with reduced toxicity. However, because it is laborious and needs technical expertise, it is dedicated to very specific applications, and results depend on the method used. Finally, lipofection and polyfection are often used because they are simple and quick transfection procedures that require low optimization. They show some reproducibility, but they present high yield of toxicity with a low to moderate efficiency. Moreover, beside to the risk of non-specific effects, these transfection methods often induce cellular activation and stress. This is why we have developed a totally new class of cationic polymers with fusogenic and pH-sensitive properties. We wanted transfection reagents that present nucleic acid shielding characteristics to lower cellular stress while promoting nuclear uptake. And as for all our transfection reagents, biocompatibility was the cornerstone of this development. This new technology was called CHAMP, standing for cationic hydroxylated amphiphilic multi-block polymer. The novel patented polymers that we have developed are thus biocompatible, ionizable, pH responsive, and bifunctional. They comprise 3 moieties for 3 synergistic notions. Concept of passing through the membrane barriers, the idea of stealth transfection, where nucleic acid is protected and masked once released from the endosome, and of course the principal asset relaying in the concept of biocompatibility. And this to promote optimized delivery system, allowing high efficiency and low cellular stress due to improved delivery mechanisms. Basically, the polymers are based on a bifunctional dual copolymer structure with 2 blocks of polymer bridged by a hydrophobic linker. And every part of the construct plays an important role in the nucleic acid packaging, delivery, and stealth effect. The first block would be dedicated to complexation, endosomal uptake, and proton sponge effect. The linker promotes self-assembly as well as fusion and destabilization of membranes. The second polymer block would offer protection in the cytosol, stabilize molecule in serum, and favor DNA uptake. And the whole idea besides this is that we can play with several iterations to finely tune the whole copolymer properties to adapt to any biological needs. The whole properties of our new cationic CHAMP polymers would allow a high compaction level of nucleic acid. So let's have a look how it works in detail. The excess of polycations at the surface of the polyplex allows attachment to the cell surface and internalization by the target cell. Once endocytosed, the nature of the polymer favors proton sponge effect. The linker is cleaved, releasing the first polymer block. As the osmotic pressure increases, the endosome rupture is favored by the exposition of hydrophobic domain. Now the second block remains bound to the nucleic acid and will reach the nucleus either directly or via microtubule trafficking. The polymer will protect and hide the nucleic acid until expression in the nucleus. Based on this new class of polymers, we have developed transfection reagents for in vitro, such as Helix-IN and VeroFect, and in vivo with BrainFectIN. Let's talk about in vitro transfection. Helix-IN is based on our CHAMP technology and comes with a transfection enhancer. The bifunctional copolymer is associated with a specific formulation developed to increase transgene expression and favor transfection. The protocol is very simple. As for any other transfection reagent, Helix-IN is mixed with DNA suspension, and after 30 minutes of incubation at room temperature, the polyplexes are added to the cells. Hib 100 is then added to the medium at 1x final. And 24 to 48 hours after, transfections are really successful, such as in the COS7 or astrocyte-derived cells. As we can see here, Helix-IN can address a variety of cell lines from CHO cells to C2-C12, Ra264, or N9 microglial cells. Of course, efficiency and intensity of fluorescence vary depending on cell type, as illustrated by the images. Hib 100X was developed together with Helix-IN and aimed at improving transfection, either in terms of percentage of transfected cells, and also mainly in terms of transgene expression. The results below demonstrate an increase in total number of transfected C2-C12 with a dramatic increase of transgene expression. Depending on the cell line, the percentage of transfected cells can also be increased. And thus the Helix-IN kit stands out from the competitors. We compared it to classic and PEI-based transfection reagents, and as demonstrated here over 5 different cell lines, Either Helix-IN performs as well as others in terms of percentage of transfection, which are the blue bars, or for the same percentage, induces more transgene expression, the orange dots. The images next confirm that the intensity of GFP is higher in cells transfected with Helix-IN than with the competitors, leading to the conclusion that this kit induces high transduction in terms of percentage and transgene expression. Most importantly, in terms of toxicity, the biodegradable nature of the CHAMP polymers allows Helix-IN to produce elevated levels of secreted alkaline phosphates, the green bars, and mainly to promote high viability index compared to competitors, the orange dots. In the same way, Helix-IN's inherent properties enable it to minimize cellular activation and stress responses, as confirmed by the reduced ROS production observed in transfected cells, represented here in orange. Let's now delve into the literature to discover what Helix-IN has helped achieve. In this letter, Helix-IN was used for genome edition in various cell lines. For CRISPR-Cas9 experiments, a guide RNA and a repair plasmid were co-transfected at the same time, and cells were selected 3 days after. The first results show that the transfection of mutant gene PDIA6 induces large diffuse signal compared to wild type, images in dark pink, and a decrease in insulin in HeLa cells or INS1 cells, as shown in the Western blots. Then transfection of PDIA6 condensates led to increase in XBP1 splicing and protein aggregation. Altogether, Helix-IN allowed to demonstrate that PDIA6 play a fundamental role in insulin folding that goes beyond classical chaperone functions, having a role in cell survival. Helix-IN was used to transfect primary human corneal endothelial cells with a nanoluke reporter vector containing segment of HLA class I promoter. Following transfection, the endothelial cells were infected with HSV1 virus to determine the mechanism by which IRF7 regulates MHC class I induction. As demonstrated here, transfection of mutant IRF7 impaired the activation of the promoter imaged by the absence of the signal. In this article, Helix-IN succeeded in providing evidence that IRF7 is essential for regulating MHC class I induction. After iterations on blocks composing the polymer and adjusting linker, we formulated a new transfection reagent, VeroFect, dedicated to Vero cell transfection. Compared to other PEI-based or classic competitors, it performs very well for transfecting Vero cells. In this example, VeroFect was the chosen transfection reagent to establish simian RNA polymerase I-driven mini-genome assay in Vero cells. Luciferase expression was significantly higher in cells co-transfected with the 3 plasmids, and the measurement of luciferase after transfection of mutated sequences was higher under the expression of L with E1497K. VeroFect transfection allowed to demonstrate the impact on viral replication and transcription. Now let's focus on our in vivo solution for brain transfection with BrainFectIN. Born from another block iteration, this novel bifunctional copolymer was developed to deliver nucleic acids in the central nervous system of small animals following stereotaxis injection. Quality controls and synthesis are upgraded to fit requirements for in vivo, and its properties allow low immunogenicity and rapid and long-lasting transgene expression. The protocol is really simple, as for Helix-IN. After complex formation, the polyplexes are delivered by intracranial stereotaxis injection. After 24 to 72 hours, we can observe transfected cells at the site of injection, for example here, in various zones of the hippocampus. BrainFectIN was extensively studied in the publication by Bisakla. Here we present a brief resume of physicochemical characterization and biological activity in vivo. The results demonstrate the capacity of BrainFectIN to complex DNA as soon as ratio 0.5 to 1, with the total complexation at ratio 2 to 1 with high efficiency. Stabilize the complexes over 3 hours, allowing researchers to perform in vivo experiment without any problem, and to protect nucleic acid from degradation as demonstrated here by incubation in 50% serum at 37 degrees Celsius. When injected in vivo in the hippocampus, the images clearly demonstrate neurons transfection both in the IPSI and contralateral zones of injection as confirmed by measurement of GFP expression. Cubic protocol after injection of complexes with BrainFectIN, and images were acquired layer by layer to reconstruct 3D images. In these videos, we can clearly observe the efficiency of transfection of neurons in the zone of injection. This next series of experiments demonstrates the functionality of BrainFectIN to transfect brain in vivo. KCC2, a potassium chloride exporter, was knocked down by transfection of a shRNA, and 10 days later, animals were injected with a convulsing agent until generalized seizures. Results show that KO animals exhibit faster seizures with a lower latency to induce them. Moreover, the expression of KCC2 was reduced by 70 and 50% in the IPSI and contralateral hippocampus. As the authors stated, this polymer-based non-viral delivery system can be used as a tool to study the functionality of the entire network, since the effect was not limited to the hippocampus. BrainFectIN also works in birds' brain for gene silencing using siRNA. BrainFectIN was complexed with siRNA and injected bilaterally into the brain of an adult Bengali finch, targeting the song promoter nucleus. The hypothesis would be that the level of the zinc transporter ZIP11 plays a role in tempo of learned song. Results demonstrated that ZIP11 knockout resulted in a significant slowing of the songs of the experimental birds, and led the authors to conclude that differences in ZIP11 levels could account for inter-individual differences in complex learning phenotype. Recently, the same team, with the work by Colqui, pursued their work in the regulation of song motor variability using BrainFectIN. If your main research focus is neuroscience, we also offer transfection reagents specifically designed for primary neurons and neuronal cell lines called Neuromag, or for microglial cells called GlialMag. These products were presented in a previous webinar, which you can watch on our YouTube channel called OZ Biosciences. Other transfection reagents from our catalog are based on the CHAMP technology and were developed for specific applications such as Polymag Premium, which is the most recent update of our famous Polymag range, and show efficient transfection and low toxicity in primary cells such as mesenchymal stem cells. XPMag, on the other hand, was developed to transfect organotypic cultures of explant such as section of the central retina. Using reverse magnetofection, it allowed to reach the ganglion cell layer and silence gene expression. What I'd like to emphasize before ending this webinar is the fact that these cationic polymer developed on a totally new technology have been cited in high-ranked journals. This success can be explained by the fact that these CHAMP polymers are non-toxic, reduce cellular stress and activation, and are really efficient. Helix-IN associated to HIB100 is dedicated to in vitro transfection, and BrainFectIN was developed for in vivo brain transfection.
So thank you for attending today's webinar. We have some questions that we will answer. If you have any question after that, you can always contact us at contact@ozibiosciences.com. And for any technical question, you can contact Cédric at tech@ozibiosciences.com. So the first question is, would you recommend Helix-In for CHO cells transfection?
Yes, but it will depend on the application actually. If you want to do a stable transfection or to genetically modify the gene of the cells and to express a transgene, then Helix-In will be recommended. But if you're using CHO cells for bioproduction, to produce, for example, antibodies or protein at large scales, then in this specific case, I will rather recommend another transfection reagent that we develop, and it will be HypeCHO. HypeCHO is a part of our Hype line of reagents that are dedicated to bioproduction, so it will be more dedicated to this application. Otherwise, for any other type of transfection in CHO cells, then Helix-In is highly recommended.
Okay, thank you. The second question is, how long does transgene expression typically last in brain after BrainFectIN injections?
Yeah, that's a very good question regarding the longevity of expression after transfection in the brain. And it provides stable and reliable expression in the brain of animals. In our study, we monitor the protein expression in the hippocampus of rat at E11, I think. And we can see the results on our websites are available. We can see a Western blot where the GFP protein is still expressed after 3 weeks after transfection. And as we can see, the amount of protein is still relatively high. So we can imagine that expression could last maybe 1 month. But of course, it depends on the animals, on the half-life of the protein, of the quantity of plasmid delivered. So the maximum time of expression is still to be determined, but let's say 1 month, to be honest.
Thank you. A third question is, can I use Helix-In to produce lentivirus in HEC293 T cells?
Yes, but it will be the same, almost the same answer as for CHO production, because Helix-In will work in HEC to produce lentiviruses because this transfection reagent is highly efficient. It presents a high level of nucleic acid compaction, and so it will produce lentiviruses and it works. But we have observed that there is no direct relationship between the efficiency of transfection and the number of our particle produced. And this is why we have developed an IV reagent that is specifically developed, dedicated to this application because it works well in HEC and it produces a lot of viral particles. So as for CHO, for any transfection experiment regarding transgene expression or transient or stable transfection, yes, Helix-In will work. But for viral production, I will recommend IV.
Another question is, what are the conditions of injection of BrainFectIN?
The conditions depends on the size of the animal, and because the size of the animal will determine the volume of injections that you can use. We're going to talk about the rat brain, for example. In this Calas publication that we presented during this webinar, they use they injected 0.5 to 2 microliter using a microinjector at a speed rate of 100 liter per minute. So it's very slow. Speed rate has to be very slow not to damage the tissue. And the crucial point was that after the injection that lasts 15 minutes, the needle was left in place for the same amount of time to prevent backflow of the solution. And so the complexes were not stuck out when removing the needle. And the major recommendation will be to consider the size of the animal and the volume once again to inject, because you don't want to go too fast to damage the tissue.
Okay. Did you see any inflammation after the injection of BrainFectIN?
Yeah, no, we do not see the presence of active gliocytes that surround the blood vessels. And we also measure the GFAP protein by Western blot, and we saw that it was really low with or non-significant when measure it after transaction with BrainFectIN. During its development, we have compared it with other competitors and other transaction reagents, and the ones that induced the most inflammatory pattern were lipid ones. And we had that one molecule that we developed that was issued from our catalog of lipids that transect very well also, as well as BrainFectIN, but the inflammatory pattern was really high. So this is why we go with our new polymers that, as we show in the webinars, they present a very low pattern of inflammation.
Okay, thank you. So we won't have time to answer everybody's question, but do not worry, we will take time after the webinar to answer everybody with an email. So please stay after the end of the webinar, because we have a quick form for you to fill in, a satisfaction survey, to see what we can improve. Thank you very much for your time, and have a nice day.
Bye-bye.
Bye.